This is the first installment in a video series about the online scientific software Benchling. In this video, we will understand the basic functions of Benchling, learn how to join an organization and navigate through Benchling's interface, learn how to organize and share your files, constructs, and projects, learn how to import DNA sequences and plasmids, and integrate the notebook function and the workspace, and learn how to export your data from Benchling. So Benchling has a couple of distinct functions that all integrate together. First of all, Benchling is an online lab notebook, which can be used as an alternative to your physical notebook. As it is cloud-based, it allows for collaboration within your lab group. Benchling is also an online plasmid editing tool, which allows you to visualize and analyze the sequence of plasmids and other DNA sequences. You can then simulate cloning experiments. And the plasmid editing tool integrates into your lab notebook. Benchling can also be used as a laboratory inventory. Our lab uses Quartzy, so I won't be going into this feature today. So now I'd like to switch over to a demonstration. So immediately after you sign up for Benchling, the first thing that you'll be shown is a screen that allows you to sign up for an organization. So I'm going to type in the name of the Wells Lab organization, which is Wells UOM. After this, after you request to join, it will take you through a couple of Benchling's features, and I encourage you to read through this and discover some of these for yourself. So this is the main Benchling workspace. On the left, we have a number of different buttons. Home shows you some suggested tasks, as well as your recently opened files once you've done so. At this point, I'm going to switch to my own account so we can talk about some features in more depth. So the next button here is Projects. This is the file manager of Benchling. Files are, by default, sorted into different laboratories. And after that, they are sorted into projects, which you can edit the permissions of individually. And then within that, you can continue to have more folders. This button up here expands the window so that you can navigate more easily and see more details about your files. You can also filter the files by either notebook or inventory. Notebook contains your lab notebook files, and inventory contains virtual molecular biology constructs. These can be DNA sequences or plasmids, amino acid sequences, or even CRISPR constructs. After the Projects button, we have the Search button. And underneath that, we have the Create button. So right now, I'm going to show you how to add a plasmid from AddGene, which, if you're not familiar, is a popular repository that the authors of journal articles use to deposit plasmids that they've used or mentioned in their papers. So let's say, for instance, that you're reading a journal article, and the article mentions that it uses a plasmid from AddGene. In this instance, it mentions it uses the AddGene plasmid with the accession number 27076. So from here, we can copy the accession number and then go to addgene.org and search for that plasmid. This will take you to the results page, which shows you details about that plasmid, including a diagram. Now, to import this into Benchling, all we need to do is copy the URL and then go back to Benchling, and I'm going to click Create, DNA Sequence, New DNA Sequence, and instead of starting a new DNA sequence from scratch, I'm going to search external databases. And then here we can just paste in the URL and click Search. And here is the plasmid. I'm going to change the name of it to IPS plasmid, just so I remember what it's for. and then I'll click Import. Now at this point, we can see on the right-hand side, we have a diagram of the plasmid. We can change the zoom level if we want. I'm going to just hide that section to make this a little bit larger. And in the diagram, we can see different restriction sites, as well as in the inside, we have the different features of the plasmid, including the origin of replication, various genes, and on the outside, we can also see various primer sites too. And if we click on any of the features in the diagram, then it will automatically move us to that particular part of the plasmid on the sequence map, which is the genetic sequence of the plasmid. Now, as all good scientists do, we should now log what we've done. Now, of course, you can continue to use your physical lab notebooks, but it may be more appropriate to store it in the cloud if it's something that you need to collaborate on, or something that you frequently want to refer to at home. So to do that, I'm going to click Create again, and then Entry and Blank Entry. I'm going to call this IPS Creation. And then here I can write about what I've done. Now, if I then type the @ symbol, I can type in the name of anything that I've imported, for example, the IPS plasmid, or just the first couple of letters, and then click on it, and it creates this link here. And this link is a dynamic link, which means that if I change the name or the location of the file that it's linking to, it will change it in your workbook too, so that nothing gets lost. Now at this point, you may want to export your work, and there's a number of ways that you might want to export your work. First of all, if you want to share any part of your notebook with somebody else without allowing them to edit it, you can click the Share button in the top right-hand corner and generate a share link. And then anyone who clicks on that link will be able to see what you've done without making any changes. The other way that you may want to export your work is if you want to use it in different contexts. If we click on the Projects button here, and then click on the gear symbol next to the project that we're in, we can click Export Project. From here, we can export the entire project as a zip archive. We can export individual entries in our lab notebook, either as HTML files or PDFs. Individual sequences that we've made, we can either export as a zip archive or as multi-part GenBank. GenBank files are files that are used by the GenBank software and other plasmid editors too. So if you've made something in Benchling and you'd like to edit it in a different kind of software, you probably want to export it as a GenBank file. The final way that you may want to export things is if you've created an image of a plasmid or any other DNA construct. You can click the PDF button here in the top right, and then you can export the diagram as a PDF. And these PDFs are vector images, which can then be exported for publication, or they can be used in vector image software editors, such as Adobe Illustrator or Affinity Designer. The next two videos in this series will go into more detail about how we would use Benchling for scenarios specific to our work, including stem cell editing. If you need more resources on how to use Benchling in general, I highly recommend going to the Benchling support site, as shown on screen, which has video tutorials for all sorts of basic tasks.